To another Google Developer Group AI Science event. It's very, very exciting to have something going on in person, hybrid. So thanks for joining online, everyone who came. And today we have the pleasure of having Priyanka and Decoding giving us a talk on decoding the melanoma tumor microenvironment with multiplex immunofluorescence and deep learning. So I'll let you guys introduce your topic and go from there. So thank you.
Thank you.
Okay, so good afternoon, everyone. I would like to begin by acknowledging the traditional owners of the land and we all meet on today. I would also like to pay respects to elder present, past, and emerging, and any Aboriginal and Torres Strait Islander people here today. So good afternoon again, and I'm presenting our work on multiplex immunofluorescence images, which has been developed to predict the immunotherapy response in melanoma patients. Our team includes Associate Professor Decoding, Professor Macquarie, Professor Rana at Centre for Health Informatics, Australian Institute of Health Innovation at Macquarie University. And we have collaborators from Melanoma Institute Australia. We have Associate Professor Wilmot, Dr. Tuba, Vizay, and Nuruddin on board. And they have been incredibly helpful for providing us the data set, these rich information data, MIF images, on which we are making our projects stand. Melanoma is a type of skin cancer that develops in the skin cells called melanocytes. And like any other cancer, it is one of the most disruptive health conditions anyone can have. A friend recently shared their family's experience 40 years ago that back then chemotherapy was considered was seen as more painful than having an alternative of not having any therapy altogether. And sadly, despite the painful sessions, the survival rates were still very low. Thanks to the advancements in medicine, we have very advanced treatments today, and immunotherapy is one of those. Traditional cancer therapies focus on killing cancer cells with drugs or radiations, but they often come with significant side effects because cancer cells mimic healthy cells and giving very low survival rates. On the other hand, immunotherapy helps the immune system, it strengthens the immune system to fight the cancer. But still, despite having a lot of successful stories, more than 60% of the patients still experience side effects, and some severe enough to stop the treatment. So what if we can predict which patient is suitable for immunotherapy? That will make lots of lives easier, as patients do not have to undergo those fatal reactions, and clinicians can focus on other alternative treatments. As a computer scientist, this is a prediction task for me, and which can be done by training a classification model on a suitable data set with labels. So here we have multiplex immunofluorescence imaging, which are a very special type of imaging that enables simultaneous visualization of multiple biomarkers within a single tissue section, revealing cell-to-cell interactions, immune infiltration, and also the spatial organization within the same tumor microenvironment, which is something very useful for downstream tasks of disease, to understand the disease behavior and treatment responses. MIF images are a bit different than the standard pathological images, which have 3 channels, while MIF images can have up to maybe 5 to 50 channels, all depending on the number of biomarkers we have used for that task. So MIF images provide us the quantitative multi-channel molecular maps that capture both phenotypic as well as the spatial organization of cells within the tissue. And this information is extremely rich and extremely biologically important, but very very hard to analytically process it, and it's very challenging to analyze this rich information and make best out of it. So this definitely requires very advanced computational models to interpret and make best out of this rich information that is provided by MIF images. So as a standard practice, usually pathologists scan the whole slide image and look for the key features throughout the image. And to perform an equivalent task using an AI model, the obvious solution is to train a computer vision model and take the whole slide image as input and predicts the output. However, this approach is quite challenging in reality, given the fact that whole slide image is actually of a very large size, sometimes up to gigapixels.
Sorry, Priyanka, can you guys hear online? Does Priyanka just need to be close to the microphone? Okay, yeah, maybe it's
how about now? Okay. Good.
Thanks so much.
So shall I start from?
I think that's fine.
That's fine, right? Yeah. So as I said, that it is not very straightforward to analyze the whole slide images because of their huge size and because of their sizes in gigapixels and the high resolution. And over that, there are usually many different types of tissue processing and man-made artifacts in the whole slide image, which sometimes dampens the model performance. And apart from that, there's a lot of information redundancy in a whole slide image, because many times the cancer, the part of the slide which tells us that this slide is has cancer is located at a very small corner of that big tissue section. So to address all these challenges related to the whole slide image, we have developed a framework, COMIL, which is channel optimization with multi-instance learning. And the study has been accepted for publication in the IEEE Journal of Biomedical and Health Informatics. It's in the press and soon it's going to be available. We have developed COMIL from the data we received from Melanoma Institute Australia. We have received almost 211 whole slide MIF images and an additional 42 samples which are reserved for testing. The image size is up to 24,000 pixels, and there are 6 molecular markers: CD8, 16, 68, DAPI, PEL1, SOX10. And each of these represent the protein that is used to visualize different sections of cells and tissues in the slide. The data set also has autofluorescence channel, which helps to estimate the tissue morphology at a very high level, like the tissue structure, the texture, and all. Each MIF sample is labeled as non-responsive, which is considered as a positive class in our study. We have 91 samples for that. And then there is other class which is responsive, negative class, with 120 samples. So COMIL is based on the multi-instance learning framework, which is a very well-established framework to analyze the whole slide images. In multi-instance learning, the whole slide image is cropped into several patches, and these patches are then input into the framework to yield the corresponding output score. And if we properly design this framework, we can reduce the input size and also potentially avoid the artifacts. We can also reduce the data redundancy and provide more room for the model treatment. model improvement. Sorry. The framework, MIL framework, has a few very important modules in it. The first one is the patch selection method, which dictates the mechanism of how the patches are going to be selected for the whole slide. And then we have a transformation function, which converts a patch into a lower morphological representation. And then we have a pooling operation that aggregates all the patch level features into a single slide level feature representation. And in the end, we have this whole slide level predictor, which converts the slide level representation into the final scope. So as a first step, we performed segmentation on our data set. And segmentation is a very important processing step, which is how we obtain clear tissue regions out of those slides which have a lot of artifacts around the main tissue region. And this segmented region is divided into multiple patches to form a bag of instances representing each slide. So this is the COML framework. And as I said, it is built on the multi-instance learning. So the input here is the patches from the whole slides, and each patch for each channel is encoded into a feature representation using a pre-trained CNN model. And we have two novel contributions here in this framework, the gated channel transformation and adaptive loss function. So gated channel transformation extracts channel-wise features capturing interdependencies while learned channel optimization. And adaptive loss function module enables the model to prioritize the training at patch level as well as at the slide level. So it enables more targeted model optimization according to the higher loss during training. So this helps improving the model capacity to learn the features at the slide level as well as at the patch level. So here, once the features are extracted, it goes to the gated channel transformation. All the features from the channels of a full slide image are aggregated, and then it undergoes the attention mechanism, and then it goes to the adaptive loss function module, and finally we get the predictions. So I'm digging a bit more into gated channel transformation, as it is our key contribution in this formal framework. So there's few mathematical operations we did here. So GCT enables the accommodation of arbitrary number of markers from 5 to 50, as I said, because the standard MIL framework just can take only three channels. So we have placed GCT just after the feature representation module so that we can combine as many biomarkers and get into the model. So we have GCT constitutes 3 submodules. One is GCE, and then mobilization and gating adaptation. And GCT, GCE enables the accommodation of all the biomarkers by summing the squared activations across the spatial dimensions. And then these activations are normalized. And then in the end, the gating adaptation is applied, which helps to model the interchannel relationships, and it just creates a kind of competition as well as a kind of collaboration among the proteins channels, so that we get the quality information, so we can feed the quality features into the model. We performed rigorous evaluation for COML. So the first set of experiments was to compare COML with other MIL frameworks. So here we see how COML stacks up against all existing MIL frameworks, and the takeaway is straightforward, that it outperforms every baseline across all metrics on both the internal and external test sets. And what's important here is the consistency. So most methods lose the ground when they are evaluated on the external test set. But it looks like that Komal maintained a clear stand here, and it tells us that the model is not just fitting in the training distribution, but it is capturing certain signals in the MIF images, which are generalized across the test data sets. So overall, these results show that the Komal offers a generally more reliable and effective multi-instance learning solution. And then the other set of experiments we did was for the ablation study. We tried to illustrate the contribution of the proposed component of Komal. So we took out each component out of the framework and tried to look at the performance drop. And it was very clear that when we took out GCT or ADL, the ones which we proposed, the performance significantly dropped. So that was another evaluation for the proposed models. And we also conducted a channel ablation study by systematically excluding each channel and evaluating the resulting performance impact. And our analysis revealed that CD8 and CD16 channels contribute most significantly to the model's predicted performance, and which was consistent with the findings that MIA had got. They also did some studies at their end, and the findings that CD8 and CD16 are the most important channels was something very consistent with their findings. And we also noted that the exclusion of autofluorescence channel led to a big fall in the performance on the external test set, showing that the autofluorescence channel has a strong role in generalizability of the MIF images, because, as I mentioned before, autofluorescence channel catches the tissue structure and the higher level morphological features of the tissue. So in order to demonstrate that GCT performance is independent of feature encoder, we evaluated it on various architectures, and we found out that ResNet 101 is doing the best. So here in this table, the base is the Komal without GCT, and the proposed is the entire Komal with GCT. And we found out that for irrespective of the feature encoder we have used, GCT has consistently improved the performance across all the all the encoders. And I just want to highlight here that the first two, Virchow and Uni, are the foundation models, and they have features from the exclusively from the pathological images, while the other architectures like DenseNet, ResNet, they are the CNN-based architectures, and they are trained on this big data set of ImageNet, which has all the natural images in it. But still, MIF images work better on ResNet 101. Now let's take a look at these images. And at first glance, I would like to ask you, like, if I tell you that all 4 of these images are MIF images, would you agree? Or do they look like they belong to the same imaging modality?
Not really, not the top two.
Oh, maybe. Okay. How about others? What do you think?
Galaxies.
Yeah.
Yes. Okay, so,
Like,
yeah, so we have all smart people in the same room. So I'll just get back here in this. So please, as I mentioned before, that virtual and uni models are actually consist of the pre-trained models from pathological images, while ResNet is from this ImageNet data set. And I found that I was expecting the Komal to work better on uni and virtual, but it worked better on ResNet. And I did more reading, and I found out that that is because ResNet is trained on the ImageNet data set, and that has a lot of space images. And we see that space images are so similar to MIF images. And I was really like fascinated with this finding, and what I could then I told myself, okay, then that means the patterns inside the cell are so similar to the patterns in the space, right? So that's how this computational model worked. So yeah, so before I turn this presentation into a spiritual discourse, let's move to the next slide. So we also produce the heat maps. These images illustrates the contributions of the proposed GCT and area to the predicted performance. And notably, the responder samples, sample 1 and 2 are the samples from the responder class. They exhibit more dense and dense regions in the heat maps compared to the non-responder samples, which are in sample 3 and 4. So this observation is consistent with established biological knowledge that responders typically demonstrate higher innate cell density where overlapping membranes of adjacent immune cells contribute to the increased signal intensities. So that also shows that COML has a potential for an interpretable biomarker discovery tool as well. So where we are heading next? So so far we see that multiplex immunofluorescence imaging has helped us to see the tumor microenvironment, to understand it to a certain extent, and deep learning models its complexity to generate the biological insights and help us to achieve a model which does produce immunotherapy response prediction successfully to a certain level, but we are going to have more clinical interpretation and more evaluation on it. So the next step is to include clinical characteristics in it and have a multimodal data and focus more on the clinical interpretations so that we can actually place this model in the real world. So that's all from my side. Thank you very much. And yeah, I'm happy to take any questions.
Thank you. Thank you. I have a question off the bat. So the framework, is it like a Python framework or like a
Yes, The code is going to be available soon.
That was going to be my next question.
Okay.
Yeah. Thank you. Anyone online ready to ask a question?
Very interested in this connection you made on the Hubble telescope at Cosmos. Is there a relationship? Because you look at the signs of everything. They have consciousness in everything. According to the science theory. So there must be some relationship to be able to match it.
Yes, and I think we did find a few papers where they have analyzed MIF images with the algorithms that they have used for analyzing the space images. So this is something very interesting and very fascinating, and it can go to all kinds of kind of perspectives. So yeah,
the only limitation is Cosmos. We can't induce drug, but even we can.
Yes. Yeah, and I think it's also MIF images. It's very hard to capture these images because they come out from a very expensive infrastructure, and having space images has its own challenges. You have to go to space, or maybe I don't know, telescope being very advanced.
Actually, there's a point there is a relationship. Then we can. One more question was the causal effect. Look at the causal. Do you feed this to a CRISPR tool or somewhere we can play with, or?
Not yet, but that's definitely our next thing that we are going to take up with the from MIA to place the model and have more rigorous evaluation on the patients. So far, as I said, that this model is evaluated on 42 samples of MIA images. So those are from test samples which have been acquired from a different set of patients. But to be in the real world, to have actual causal effect, I think we need more rigorous evaluation, which is the next part of this study.
Are you actually, oh, sorry.
Why I'm asking that is because it can predict, we also can prevent it. Putting a monitor, say like a gadget, thinking now in a startup phase, I can create a gadget and monitor, and it can predict things. I can put a software predict this is possible for you to catch cancer because these symptoms are give the trend analysis. Right. So prevention is the real cure rather than.
I think yes, that's a very important point, and but it is possible, and I think I just saw this kind of thing in one of the posts on LinkedIn. The conference was held in Germany, somewhere in Germany. So there was a big image on they were talking, like all the experts are talking about it, that they have already developed the model that based on my, let's say my pathological images or my clinical data, they can predict whether I'm going to have, this kind of disease in future or not. So it all depends upon the data set actually to have. And
But you got a scope to work on that?
No.
Can work on this area? Or?
Actually, this is something different, like, because as I said, we have collaboration with MIA, so we have common goals. And, current goal is to predict immunotherapy response.
Right.
All right, good stuff. Should we plug? Oh, sorry.
Oh, yeah. I was just saying, how do you actually grab the MIF images? Is it like a patch, like the images, the input?
I think they have, the tissue sections are grabbed from the human body. And
Also,
then,
like a physical.
yes, so it goes into a lab, undergoes that staining, and then they have these very advanced machines.
Yeah.
And yeah, that's how they do it.
Okay.
That's a very important and I think a very elaborate process that happens at the backstage for us.
Yeah,
And,
yeah.
we are working with the AI thing. So
Would it matter if it's just in one section of your body? This is probably in science, but if it's just in one section of your body, would it matter that the cancer is like somewhere else? Or is it all the same, like consistent?
I'm not an expert in that. Maybe
Yeah.
a clinician can suggest that better, but as far as I know, they always look for the skin lesions on the body.
Yeah.
And then I think they extract that part and put it under microscope, stains
Yeah.
it.
Like for this specific project,
Yeah.
we are looking at, the melanoma patients who has already progressed to the advanced stage.
Yeah.
So these are the stage 3, stage 4 patients. So the marker of that is like they already have the metastasis. So all the tissues are actually taken from the lymph node.
Oh, yeah.
And have two question please, like information talk. First one is about data sets. Got 211 samples.
Yeah.
I'm not sure it's enough. Do you collaborate it overseas to get more data on top of the melanoma Australia? To talk about it, open sources.
Yeah, I mean, this is a private data set, and we do not have any collaboration overseas about it. I mean, we are not aware if any lab has this kind of data set, which is specifically for immunotherapy response prediction, because we may have a lot of MIF images all around online, but it depends on what labels do they carry, because we are looking at the MIF images which have proper labels of responsive and non-responsive to immunotherapy. So, yeah.
Second one is about translational research.
Sorry.
My translational research. I heard about Harrison AI.
Yes.
They have the software analyst. It's called pathological diagnosis. Have you thought about in the future, like to translate from research to commercialization? Would you like to work with, collaborate with Harrison AI?
I mean, this actually a project, we co-developed with MIA. I guess, essentially, our goal is to translate this into a proper health service so that people can benefit from this. So any help, from, commercialization team, from industry partners, if MIA has interest, we'll be happy to.
Thank you.
Can I have one quick question? So the common framework outperform the other compared to the others. So I want to ask, like, what is the thing that makes the commune model like better than the other models from the data from the how to say the one that you show on the table, the data.
Yes, that's a very good question, actually. Yeah, so see, I mean, based on the evaluation, so as I said that COML did compare COML with other multi-instance learning frameworks, which are based on attention mechanism, transformer-based MIL, then we have prototype learning thing. So these frameworks have been developed for some specific task, like let's say feature refinement for a standard pathological image. And we designed COML specifically for multiplex immunofluorescence images, taking care that they have like, they may have 5 to 50 channels. So I think the more focus on that problem part of computation of MIF features that helped COML to stand out than other MIL frameworks, because when they were those frameworks were developed, they had different problem area in their mind.
So is it like fine tuning?
Oh no, it's not fine tuning, because we did introduce 2 modules, as I said, GCT and ADL. We did introduce 2 modules in the standard MIL framework. So all the MIL frameworks that I mentioned in that slide, they are also based on the same MIL standard MIL framework with little bit one or another module in that, or maybe different purpose like feature. And also they are evaluated on different type of images. They are evaluated on H&E stained images.
I see. Thank you.
Just like would like to add a few more comments on this project. So I'd like to emphasize on the clinical relevance of this colonial framework. So for every project our team is working on, we are trying to answer a clinical question. So in this specific project, we try to predict if the patient will respond to a specific immunotherapy. But for melanoma patients, I guess there are more than just one immunotherapy available to them. So it's kind of related to your question. So if we can predict these patients will respond to this treatment, then it will be a good option for the patient. But if we choose a different immunotherapy, perhaps that would lead to severe consequences in the patient. So it's kind of trying to predict what will happen to the patient so that we can optimize the treatment for patients. Yeah. And just to give some context. So based on the clinicians' experience and choosing the chance of choosing the right therapy for patients is about 50 to 6%. And there are also other teams looking at the clinical data and the genomic data. So you can consider these as different input, but they can be used for the same task. But we are looking at the imaging input. So if you look at the clinical and the genomic data, I think at the moment they achieve AUC of about 60 to 70%. But because the MAF image, they provide cellular level the rich information about the tumor microenvironment. So that's why I guess the imaging modality model achieve up to 80% AUC. So what you can imagine is if we combine all 3 clinical, genomic, and imaging data, perhaps they can further improve the accuracy. And technically, and these the main contribution of this work, as Priyanka mentioned, is we are kind of the first to develop a multi-instance framework to handle the multi-channel image input. This is not feasible with the typical MIF frameworks for the AIHI stained images. And we focus on we utilize the interdependency between different channels so that it can achieve better performance than the typical and then other modes. And I'll just say like this, one example how kind of a good example showing our team's approach. We always start from one clinical problem, and then we translate it into a computational problem, and then develop like models to solve the computation, and then at the meantime, we solve the clinical problem. So I guess like I can move to my my talk. This is actually related of a side project from Priyanka's project, and this work is actually led by one of our PhD students. His name is Sunadi. I don't know if he's online. So when we were working on the melanoma project, we noticed this, for these multiplex immunofluorescence imaging data, apparently it is more complex compared to HNE or other immunohistochemistry images. And the stain failure rate is up to 25%. So that means like 1 out of 4 slide section underthrough these multiplex immunostain will fail. So then I think about that, like if it takes a lot of time to collect up to like 100 patient data, 50%, like 50 patient data are unavailable, that would be a big loss to us. So we think like if there any way we can kind of compensate the failure rate, then we come up with a solution. So why not try to use the generative AI, to impute, or this is a typical like image generation methodology to impute the stains. So there are a few typical uses of the stain imputation or the generative AI in this case. So for the images with the missing or failure stains, we can use generative AI to restore the images. And for the images does not have a specific stain, we can create or synthesize the stains. Imagine if we have a multi-center project. Different centers, they have different stains, but they want to work together on the same project. Then we can use the generative AI to synthesize the missing magnetic for one center, and then so that can match the other centers. So these are why we think we should we have why we came up with this idea to use generative AI for stain integration. And earlier this year, we actually started working on this project, and we so far have actually 2 papers. One is published and one is submitted, but we're still working on this. And these series of projects is led by Sunadi, the PhD student, co-supervised by Priyanka and myself. Okay, so the MNF imaging is actually not quite accessible compared to other histopathology images. So there are not many data sets available, and that's but it is emerging. And the papers on stain imputation for MNF images should really there are not many papers on this. So we reviewed the literature and we found just like 2 studies focusing on addressing this problem. And the first one is called Maxima, and the second one is called MS. So these existing method models, they either are based on, for example, the Maxima, it is based on a GAN model. So it's actually a conditional GAN model. So they use the stain image to generate the target stain images. But this is like a fixed input and fixed output approach. It's not quite flexible. And then later on this MAS framework, it extends the maximum framework by allowing flexible input but the output is still like by allowing the fixed input, but relaxed on these output selections. So this is a fixed input and a flexible output approach. And this one is based on the master encoder approach. So by the way, there are a couple of widely used generative AI models, like a GAN model, so we're all familiar with. And this master encoder is number one. And diffusion model, of course, this is I think this is the art. All right. So we based on these two works, the Maxim and Nice, and our team we developed two other frameworks. The first one is called SMERF. I'm going to give more details about this data. And the second one is called Assuming. Priyanka has already described the dataset we used. So skip this part. For the first framework we developed is again based on a GAN model. This framework is built upon the Maxim framework, but instead of just using the fixed input, we try to mimic the missing modalities, missing input in this scenario. So we add the channel-wise, we add we remove patches from host images, so that we enforce the model to learn to perform more robust and challenging cases. So this is a kind of a strategy similar to the dropout training strategy, so that model can predict what to impact and predict what the content should be, even just with limited input. And that one was published at the ESB conference this year. This is showing a few examples of the SMERF performance. And we can, the one in the middle is SMERF outputs. So we have noticed that a few problems with the SMERF. It produces blurred images, and maybe it's not quite clear here, but it's because it uses patch as input, so you can see the boundary. It's kind of not quite natural. This has this patchy effect, and then the imputation is not perfect. This is kind of inaccurate in some cases, and despite of these drawbacks of a SMERF, just provide higher flexibility on the input. And then later we developed these axial memo framework, and this one is I think the limitation of SMERF is mainly attributed to the GAN model. Then we choose MAE multi-masked autoencoder as the backbone for the new model, and we also use the same strategy to mask certain channels or certain pictures from the images, so that to enhance the robustness of the model. So this model is flexible in both input and output, and it's also more efficient compared to training again. So I'm going to skip the technical details just to show a few examples. So with this imputation of this axial memo, if we have more available biomarkers for the imputation, it performs better. But actually, all models perform similarly. But the main advantage of the axial memo is that, so if we only have limited input biomarkers like AIHI or autofluorescence, it can still produce reasonably good results. So this I think it's the one on the left is the one is the results when we use more input channels, and the column on the right other results, we use only RPN autofluorescence as input. So these are known as the non-antibiotic stained images. And for some, maximum, for example, the existing models almost completely fail in these limited, input scenario. Okay, and in terms of we evaluated the image quality based on structural similarity metrics, Pearson correlation coefficient, and peak single to noise ratio is typical image quality metrics, and outperforms better than existing methods. All right, so I think that's just a quick a presentation on the extension on Priyanka's project. But this is also, I would like to also quickly showcase a few other projects we use generative AI for synthetic medical imaging. And so Dr. Thomas Kong from our team, he developed a model based on GAN for stain normalization. So if we have images from multi-centers with varying stain color, then we can use this stain normalization to harmonize the stain colors. So it can improve the downstream diagnostic performance. I also have a project led by another PhD student, Wen-Jin Zhong. And his project is on using diffusion model to synthesize gray MRI images. And Xingnan is also involved in another project to virtual stain the unstained images. And at the moment, we are looking at the HLE stained images. And then that's the invitation. That's the focus of my current talk. All right, so that's the last. I would like to just to introduce a bit about our team. So we are the AI-focused civil medicine team within Center for Health Informatics, which is the center at Macquarie University. And our team focus is to integrate AI into clinical practice to improve the patient outcome. And our focus, as you can see, is on cancer research. And we also address practical challenges in developing and implementing AI. For example, our models try to ensure the data privacy and reduce the computational resource demand and ensure robust performance in different healthcare settings. So then, last, I would like to acknowledge the funding bodies. So our projects are supported by Australian Melanoma Research Foundation, AIHI, Melanoma Research Alliance, Cancer Council New South Wales, and Cancer Institute New South Wales. And we also would like to thank Melanoma Institute Australia patients and their families for their contributions to our study. Ongoing support from our colleagues at MIA and the Charles Perkins Center, University of Sydney, Macquarie University is also greatly acknowledged. And support from the Clearbridge Foundation, as well as our colleagues at MIA and the Royal Prince Alfred Hospital is also greatly acknowledged. Thank you.
Thanks very much. Are there any questions? I always have questions, so I'll hold back. Anyone online, feel free to ask a question as well.
All right. So I saw like you mentioned about the high failure rate of 25%. Is it due to like data limitation or the image limitation?
Well, I don't know, again, I'm not a clinician, I'm not an expert in this, but just from literature. I think the main reason for that, for the higher failure rate, is because this is like a very complex staining protocol. So rather than just like a stain the same tissue section once, you do stain multiple times. And in between, you also need to, watch out the previous stains. I think because the stain restaining may cause the higher, failure rate.
For the models that you mentioned about like Asimimo, Maxim, something like that, is it used to like generate the image, like replicate the original?
Well, for the GAN model, no. The GAN model, they use, they fix the input and then they generate the targeting stain. So the output and input are not necessarily the same. But for the MAA mask encoder, because it's autoencoder, so we try to reconstruct the input images. So they use different methodologies. And I guess like for diffusion, it's also like a different. By the way, there are few extensions of our current stain imputation project. So we are testing on optical data sets, and we are also like testing diffusion models for the same task. The problem is like for training a diffusion model, it's very complicated. Require like the computational resources.
It's very slow as well. I mean, for just one experiment, it takes hours.
What are you guys training on?
Study and
All right, yeah. That's a big computer.
yeah, but still it's like it's so heavy. The computations are so heavy.
I have a more general question, I guess, related to the commercialization and everything. What's the like you have a good collaboration with MIA. What's like the time from research to the actual like people using this for helping patients, or what's that pathway like? Tell us a bit about that.
Yeah, so I think like it's a really good question, and this is also what we always ask our clinical collaborators. So we don't want just to stop by publishing papers. We want to see this can be used by clinicians to improve patient outcome. So lucky enough, so within MRI, they have I think it's called personalized immunotherapy platform, and this is a program led by our collaborator, associate professor James Wimmel. And as mentioned, we have already looked at the clinical model, the genomic model, and now we are working on this image model. So through there, they call it like PIP program. So their PIP program, they have already like rolled out the two models, and hopefully our model will be the next. And they also running multiple trials within AIHI. So it's kind of a very streamlined process from the research to the testing to patients.
In quantum, we use the error factor to find. Is any error factor used to strengthen the data, or?
No.
Can you elaborate like error? This is new to me.
Quantum, because of the qubits, they lose their entanglement and those properties because of noise and atmosphere environment. Right? So we use an error factor logic to make it so that it gives you a predict a nice 99% results.
Or to compare.
So do you have some baseline like?
Yes. So for these models, we had original images. We have like full quality 211 images, or in fact 211 plus 42 images. They are all quality images. But we just formulated this problem because in other data sets we have like missing channels, or we are just like in future, as I said, they can have few tens of channels, and there is a probability of missing out the channels. So we just formulated this problem and we compared the synthetic images with the original image quality. And
When you input at a confidence level of say 75% image, the output will be 65%.
Hmm.
So what level of confidence you this input is?
It's kind of like the area under the curve and everything, isn't it? Like those an F1 score on them.
yeah, we have actually for the stain imputation task, we had other measures, Pearson coefficient, then MCC, I forgot the full form.
Some sort of.
Yeah, there are metrics specifically designed for synthetic image evaluation.
So that stands at 100% of.
No, it's not. I mean, it is, these are good metrics. I mean, we got good results, maybe I think they're in 80s or somewhere, but not 100% yet. And there's also a question on that whether we can use synthetic images for the downstream tasks or not. I mean, it's still another evaluation.
Like that's another extension of this stain imputation project. Xing'an is using the imputed or synthesized images as input and to segment cells from the images and then compare the cell segmentation performance from the synthesized image with the real image. And there's still like a room for improvement. So at the moment, I think like the performance is still like a big gap.
Right.
All right. Thanks very much once again, and thanks very much again, everyone, and let's see you all again next time. I think we'll log off online and